This is an OCR A-level biology video on Unit 2.3 Nucleic Acids. So in this video I'm going to be looking at DNA replication, transcription, translation, and the general structure of DNA, which is where we are going to start. Okay, so DNA is formed of nucleotides. So nucleotides, they're biological molecules. They have phosphate esters of pentose sugars. They have a nitrogenous base attached to the carbon 1 of the sugar residue, and a phosphate group attached to either carbon 5 or carbon 3. Okay, they're monomers of nucleic acids, forming DNA and RNA. The only difference between DNA and RNA is RNA has the ribose sugar, whereas DNA has a deoxyribose sugar. Okay, so this is the general structure of a DNA molecule. As you can see here, this circles our phosphate group, then we've got our deoxyribose sugar and our nitrogenous base. Okay, so this is how it's formed. It's formed two opposite strands, one strand's facing this way, other strands upside down, forming, so that we call them anti-parallel strands, and our nitrogenous bases bind together by hydrogen bonds, and our phosphate groups bind to our carbon 5, which is here, and carbon 3, which is here. So then we'll see phosphodiester bonds. So let's just ignore the ATCG bit for 1 second. So I'm just going to speak to you about AMP, which is monophosphate, ADP is diphosphate, ATP triphosphate. Okay, so they are phosphorylated nucleotide bases, and the only difference is the amount of phosphates they have within them. And this is what I'm drawing here, is what a phosphate looks like. So AMP mono, ADP diphosphate, and ATP triphosphate. So when you hear those molecules, now you have an understanding of what they look like. So now let's go back to DNA itself, and it's bonded like this. So adenine to thymine, cytosine to guanine. There is no exceptions to that. Okay, adenine will always bind to thymine. It will never bind to C or G. Cytosine will always bind to guanine. It will never bind to anything else. Okay, the only actual time that changes is when we're dealing with RNA, and that causes the T in RNA. Thymine does not exist. It's replaced with uracil. But their molecules are very similar. They both have the same sort of functions. They both bind to A, and they are both pyrimidines, which I'm about to tell you about. So last thing to look at with DNA, it's hereditary material, anti-parallel strands. It forms a double helix, which, if you don't know what a double helix looks like, I'll just draw for you one on the screen. So that is what a double helix is, and you have your hydrogen bonds between the bases in the middle of that. Okay, so as we mentioned before, we have phosphodiester bonds between our phosphate group and our sugar residue, and then you have hydrogen bonds in between our bases, but the number of hydrogen bonds is dictated by this. Okay, so purines have 2 rings, pyrimidines have 1 ring. So if you don't understand what I mean by that, here I've drawn a purine and circled it there. Okay, so a purine has, this is its nitrogenous base, it's got the base and a second ring attached to it. Okay, so it's sort of like a double base, but a pyrimidine will just have this first nitrogenous base. Okay, it's just 1 ring, and you need to remember which ones are purines and which ones are pyrimidines. Purines, the word purine does not have a Y in it, so they are the 2 bases without Y's. So adenine and guanine, A and G are purines. Pyrimidines contains a Y, so all the bases with a Y in it, cytosine and thymine, are pyrimidines, and let's remember that uracil, which is our RNA replacement for thymine, although it doesn't contain a Y, we're just going to remember it's the same, pretty much the same as thymine, but in RNA, so it is a pyrimidine too. Okay. One last thing to look at is the actual hydrogen bonding between the two. So A to T, adenine to thymine, forms 2 hydrogen bonds, whereas cytosine to guanine forms 3 hydrogen bonds. And I'm going to draw that here. So these are adenine, thymine, guanine, cytosine, cytosine, guanine, adenine, thymine. Okay, and AT is 2, CG has 3 bonds. There's another CG with 3 bonds, and there's another AT with 2 bonds. Okay, 2 hydrogen bonds between the molecules. Right, so that is pretty much all of DNA. Okay, but one last thing to look at is what on earth am I talking about when I'm saying 5' and 3'? Okay, so basically 5' is the phosphates binded to the 5th carbon at the top of the chain. So the top one is 5', and the very bottom we've just got our 3 carbons, so that's 3'. So this chain we say is 5' to 3'. But this chain is the upside down version of that chain. Okay, the top carbon is our 3 carbon, and the bottom one is our phosphate group bound to our 5th carbon, carbon 5. So we say that is 3' to 5'. Okay, you've just got to remember DNA is always replicated from 5' to 3'. Okay, it always goes that way. But you can just hold that in your memories for a while, because before we do DNA replication, which I consider harder than transcription and translation to understand, we're going to look at transcription and translation. Okay, so this is a picture that my teacher gave me when we was learning transcription and translation, because it is not easy to get a grips of. But how does DNA code for polypeptides then? So I've spoken to you a little bit about RNA. It's got a ribose sugar, nitrogenous base uracil replaces thymine, and it is a single-stranded polynucleotide. So whereas DNA is double-stranded, RNA is always single-stranded. It is a shorter polynucleotide, and it's got 3 forms of RNA. You can have messenger RNA, which is mRNA. You have transfer RNA, tRNA, and ribosomal RNA, rRNA. Okay, and let's compare that to DNA then. So DNA is a degenerate code. A triplet of bases forms a codon, and each codon codes for a single amino acid. Okay, and that's important to understand later where we're looking at translation. So codon is 3 bases, and each codon, so each 3 bases, codes for an amino acid. Let's look at transcription. Transcription takes place within the nucleus of cell or the cytoplasm of bacteria. So the genome zips using DNA helicase. Hydrogen bonds between complementary nucleotide bases break, and RNA polymerase enzyme forms temporary hydrogen bonds between RNA nucleotides and complementary DNA bases. Okay, a length of RNA which is complementary to the template strand of the gene is produced, and that is called pre-mRNA, so pre-messenger RNA, which uses uracil instead of thymine. So we've spoken about RNA, and that is our coding strand. Okay, so pre-mRNA is the coding strand. The mRNA passes out of the nucleus through a nuclear envelope and attaches to a ribosome. Okay, so by the end of transcription, we've unwound our DNA into 2 single strands. So breaking the hydrogen bonds, we formed a molecule of mRNA, so complementary to the DNA. We formed that mRNA, and then we've attached that mRNA to a ribosome, and that ribosome is important when we look at translation. So our coding strand is now attached to our ribosome, and translation can begin. So translation involves tRNA, so transfer RNA molecules. They are clover-leafed in shape, as you can see there, and they have bound to them an amino acid at one end and an anticodon, so which is a complementary codon to a codon on the mRNA. That's called our anticodon. That is on the opposite end. So what happens is tRNA molecules bring their amino acids and bind to the complementary codon on the mRNA molecule. Ribosome with the mRNA attached, when the 2 amino acids are adjacent to each other, forms peptide bonds between them. So to form these peptide bonds, ATP energy is needed, and then you can form a long polypeptide, which is our protein. So polypeptide is lots of amino acids together in a chain, and then our polypeptide can be transferred across the same cell to our protein packing organelles, so like our Golgi apparatus, our smooth endoplasmic reticulum, rough endoplasmic reticulum, and they can be packaged and their 3D shape can be formed, etc. Okay, so tertiary structure. That's something else I'm not going to go into too much detail about. But if that was a bit fast for you, let's look at this diagram to break down what's happened. So our tRNA has arrived at the ribosome. It's got this anticodon, so a specific triplet code, which is complementary to a codon on our mRNA, which is in the ribosome. So it attaches to that, forming hydrogen bonds between the tRNA and the mRNA. The tRNA is carrying with it an amino acid. So when the temporary hydrogen bonds form, the amino acid is then bound to the next tRNA molecule's amino acid in the chain. Okay, so a second tRNA attaches, its amino acid binds to the other amino acid by a peptide bond, then they keep moving down the chain, new tRNA molecules come with anticodons, and it just keeps repeating. So when a new tRNA molecule binds, its amino acid forms a peptide bond with the previous amino acid in the chain, and eventually we get a growing chain of amino acids, so a polypeptide chain is formed, and that can go off for post-translational modification. So now let's look at DNA replication, which I consider slightly harder. So DNA replication is during the interphase of cell cycle, and we call it semi-conservative replication. So we've got 2 DNA molecules, half old and half new. So that's our end products. So we go from 1 DNA into 2. The original DNA ends up in the new DNA, if that makes sense. You will understand that at the end. So our DNA unwinds using gyrase enzymes, and that's what this diagram's illustrating here. So we've got our DNA helix. It's unwound by helicase enzymes, forming a straight double chain of DNA. Okay, so that's activated by gyrase enzyme. Then we have the DNA unzip. So it's unwound by gyrase enzymes, so that means the double helix turns into a double strand. Then it's unzipped into 2 single strands, and that uses DNA helicase. So let's represent that there. We remove the hydrogen bonds, that is unzipping, and that's done by DNA helicase. DNA polymerase catalyzes the addition of new nucleotide bases from 5' to 3'. The leading strand is synthesized continuously, whereas the lagging strand is discontinuous fragments joined together, catalyzed by ligase enzymes. So the discontinuous fragments we call Akazake fragments. Right, so this is what's happening. So we've got our double strand of DNA, okay? It's been drawn on very slowly for you just to make it a bit easier. So we've got a double strand here. And in between our double strand we have hydrogen bonds, which is shown in red. Let's speed this bit up slightly. Right, and then that gets unwound, okay, by gyrase. It's unzipped by DNA helicase, okay, so that's become unzipped. And then we've got our two strands. We've got our 3' to 5', which is this one, so 3' to 5', and we've got our 5' to 3', okay. The 5' to 3' is our leading strand, the 3' to 5' is our lagging strand, and the part in the middle that's being constantly unzipped is our replication fork. So DNA helicase is unzipping at the point of replication fork. We've got our leading strand, so DNA polymerase here continuously adds on nucleotide bases, so this strand's being fed through, it keeps moving that way, so let's draw an arrow so you sort of understand that. This strand is being fed through our DNA polymerase, which continuously adds nucleotide bases, and that happens on the leading strand, okay, so from 5' to 3', we have DNA continuously being added, and that will produce the strand, which is obviously anti-parallel opposite 3' to 5' on the strand. So that is our continuous addition of nucleotide bases on our leading strand. Then we've got our lagging strand, okay, which is the harder one to understand. So at the replication fork, we've got our DNA polymerase again. However, as we know, we code for the mRNA from our 5' to 3', but this is the wrong way round. The strand's being fed constantly through. Remember, we're constantly unwinding this, and these two strands are being fed through our DNA polymerase enzymes, which remain at the replication fork. They do not move. It's the DNA which is moving through them. Okay, so as DNA is moving through it, it is fine for the 5' to 3' end, so we can just continuously move it through. But as we know, DNA can only be coded for from 5 to 3. Therefore, we have an issue on our lagging strand as it's being fed through from 3 to 5. So what happens? Primer annealed by action of DNA polymerase, and the DNA keeps being fed through. So DNA sort of loops up round here, and no hydrogen bonds are formed, no mRNA attaches. But at this point we have a primer attach, okay, it anneals, and this polymerase enzyme can cause the hydrogen bonds to form between the complementary nucleotides from the 5' end to the 3' end. So it does it in short fragments. So our RNA polymerase stays fixed at the template replication fork. However, it codes from the 5' to the 3' end because of addition of a primer, and then both these two strands are fed continuously through even further, and it will repeat itself, okay, again. So this bit that hasn't been coded for moves here, and the primer anneals, and the DNA is coded, okay. Originally we've got one strand, okay. Unwound, it's unzipped. The leading strand is continuously added nucleotide bases to by DNA polymerase. The lagging strand is has nucleotide bases added to it in fragments, okay, called Akazake fragments, and eventually we get two double strands. Okay, one here, one here. The DNA is completely unwound, and then it's wound into the helix. So then we have two DNA helixes produced at the end. So that is DNA replication. In the description, I'm going to link a video I watched on DNA replication that shows an animation of this. It's only 1 minute 20 seconds of watching, but it really does help you understand what I was trying to illustrate to you here. It's so much easier if you can see your strands moving through the DNA polymerase enzymes. Okay, so I'll put that in the bio. Really helpful video to watch, and it will help you understand it, hopefully. Thank you for listening. That was nucleic acids. Remember to watch that video on DNA replication. See you next time.